NF-κB pathway involves nuclear factor kappa B proteins, a family of highly conserved transcription factors. Once activated, NF-κB pathway regulates the transcription of various genes, which leads to the control of important cell processes including cell survival and programmed cell death. Key players of the pathway include NF-κB protein family, signal adapters, receptors, inhibitor of kappa B kinases, and inhibitor kappa B family. Nuclear factor kappa B family is shown here. These proteins can be classified into two classes. Class 1 includes P52 and P50. P52 was originally P100 with ankyrin repeats. It is converted to P52 upon degradation. P50 was originally P105 with ankyrin repeats. It is converted to P50 upon degradation. Class 2 includes C-rel, rel-A, and rel-B. Rel-B and P52 forms a dimer in non-canonical pathway, which is then transferred to the nucleus. In the canonical pathway, rel-A and P105 forms a dimer. Upon the removal of ankyrin repeats, it is converted to P50 and rel-A dimer, which can now bind to the inhibitor for inactivation of pathway. IKB family is shown here with their ankyrin repeats. In canonical pathway, IKK alpha, IKK beta, and IKK gamma forms a kinase complex. Meanwhile, in non-canonical pathway, two units of IKK alpha join to form a complex. Similarly, in canonical pathway, inhibitor kappa B is the primary inhibitor. But in non-canonical pathway, P100 protein is the primary inhibitor. Let's focus on the role of IKB and IKB kinase in the NF-κB pathway. In the cytoplasm, dimerization of NF-κB proteins P50 and rel-A occurs. An inhibitor kappa B binds to this dimer, preventing it from entering the nucleus. Enters IKB kinase. It phosphorylates IKB, subsequently releasing the NF-κB dimer that can now enter the nucleus and act as a transcription factor. The phosphorylated IKB itself is ubiquitinated and degraded by the proteasome complex. NF-κB pathway is regulated in two ways, namely canonical and non-canonical pathway. Here we will be showing both pathways side by side. As you can see, receptors of canonical and non-canonical pathway differ. Therefore, the ligands for both pathways will also differ. Once the ligand binds to the respective membrane receptor, adapter proteins will be recruited to the cytoplasmic domain of the receptors. Here, TAC1 is recruited in canonical pathway and NIC is recruited in non-canonical pathway. Adapter proteins recruit the IKK complexes. Keep in mind the difference between the IKK complexes in the two pathways. Simultaneous with the recruitment, IKK beta is phosphorylated in canonical pathway, while both units of IKK alpha are phosphorylated in non-canonical pathway. As we already know, IKK complex then help in the degradation of inhibitors attached to the NF-κB dimers via phosphorylation. The ubiquitination and degradation of inhibitors in canonical and non-canonical pathways is shown here. Notice, while the inhibitor of non-canonical pathway is degraded, P100 precursor is converted to P52 after processing. The free NF-κB protein heterodimers are now able to translocate inside the nucleus for transcription of genes. In canonical pathway, NF-κB heterodimer may interact with certain co-activators such as P300 or histone diacetyl transferase complex. Transcribed genes will then aid in certain cell processes. We will now talk about the significance of NF-κB pathway. NF-κB transcription factors regulate the maturation of dendritic cells. Through the transcription of IL-12 and IL-23 genes, NF-κB promotes differentiation and activation of T cells. The pro-inflammatory function of NF-κB has been studied extensively in the macrophages. Both macrophages and dendritic cells aid differentiation and activation of T cells. P50 and P65 heterodimers also regulate the neutrophil recruitment to the injured sites to phagocytose apoptotic cells. NF-κB regulates the production of adhesion molecules for cell adhesion and invasion and metastasis in cancer cells. Anti-apoptotic factors are also regulated for cell survival. Cell cycle regulators are also promoted by NF-κB transcription factors for cell survival and proliferation. Cytokines and chemokines are also regulated by NF-κB pathway, which promote proliferation, inflammation, and angiogenesis, respectively.
Earlier in the video, all the mechanisms that activate NF-κB signaling pathway have been described. But what happens when NF-κB pathway gets disrupted due to mutation or any environmental factor? So let's just dive into this to get a better idea. As explained before, NF-κB activity is stimulated through signaling from extracellular stimuli. In resting cells, what happens is that the inhibitor of Kb, as you can see over here, this inhibitor of Kb sequesters NF-κB in the cytosol, thereby preventing its nuclear localization and transcriptional function. But cytokines or PAMP ligation of cell surface receptors results in the activation of IKK complex through phosphorylation. This IKK complex is a multimeric complex of 3 subunits. The 2 are the catalytic subunits, which include IKK alpha and IKK beta, and the other one is the regulatory unit, which is IKK gamma and NEMO. When IKK is activated via phosphorylation, so it brings about the phosphorylation of IKB molecule as well, and it promotes the degradation via proteasome ubiquitin system and releases NF-κB, which translocates to the nucleus and promotes transcription of genes. Extensive research has defined IKK beta and NEMO as essential mediators of inflammatory and immune responses, and loss of IKK beta and NEMO functions is not compatible with life, and it leads to diseases. Overactivation of NF-κB is one of the primary causes of diseases, and this is all because of the constitutively nuclear NF-κB. Okay, so IKB mutation, translocation, amplification of NF-κB subunits, viral infection, and constitutive IKB kinase activity leads to constitutively nuclear NF-κB. So, in most cells, NF-κB activation is transient. We know that when NF-κB is translocated into the nucleus, it brings about the transcription of some genes. So, it also induces the transcription of IKB alpha, among other target genes. And we know that IKB alpha is a catalytic subunit of IKK complex. So consequently, NF-κB activation normally results in a negative feedback loop. So the newly synthesized IKB alpha leads to the re-sequestration of NF-κB in the cytoplasm. Although much of the NF-κB remains sequestered in the cytoplasm of primary B cells and B cell lines, these cells also contain constitutive levels of nuclear NF-κB that can be increased upon stimulation, and overactivation of NF-κB results in conditions like inflammation, inhibition of apoptosis, proliferation, indirect tumor suppressor inactivation, and these results in diseases like inflammatory diseases include arthritis, asthma, and others. And also it results in cancers like Hodgkin's lymphoma, breast cancer, and others. Moving on to the diseases that may result due to pathway disruption. So the 2 diseases that are going to be discussed over here includes hypohidrotic ectodermal dysplasia and incontinentia pigmentae. So let's begin with hypohidrotic ectodermal dysplasia. It is a rare inherited multi-system disorder that is mostly inherited as X-linked recessive genetic trait. But what are the causes, the genetic causes? The genetic causes include mutations in genes like EDA, EDAR, and mutation in IKBKG, which is the gene that encodes for IKB gamma kinase, which was previously known as NEMO gene, and mutations in NEMO are not compatible with life and leads to several genetic disorders. And mutation also include in the gene that encodes for IKB alpha. Symptoms can be many more, including immunodeficiency, depending upon the gene mutation, but main characteristics include hypohidrosis, which is the reduced ability to sweat, hypodontia, which includes missing teeth, and the third one is hypotrichosis, having fine sparse hair. Here is the diagrammatic representation of the pathway that leads to hyperhidrotic ectodermal dysplasia. Mutations in gene that causes this genetic disorder will describe which pattern of inheritance it will follow. The mutation in EDA gene, which is the first factor gene of the pathway, leads to X-linked HED. Mutation in EDAR results in autosomal recessive HED, while mutation in EDAR-ADD results in autosomal dominant HED. The ectodisplasin pathway regulates NF-κB signaling through a key regulatory protein, IKK gamma, which is also known as NEMO. Hypomorphic mutations that affect this IKBKG function also cause a closely related disorder known as hypohidrotic ectodermal deficiency with immunodeficiency. Any one of these mutations ultimately change the targeted gene to be transcribed, and this also results in the formation of defective proteins. Complete loss of IKBKG function causes prenatal lethality in affected male, and it causes the allelic disorder incontinentia pigmenti in females. And this disorder is discussed next, so let's move on. Incontinentia pigmenti is a severe X-linked genodermatosis with an incidence of between 1 in 10,000 and 1 in 100,000, and it almost exclusively occurs in females, as male cases die in utero before the second trimester. IP is characterized by certain distinct dermatological stages that begin within 2 weeks after birth with blisters and an inflammatory response. The first stage is vesicular stage, where there is massive eosinophilic granulocytes infiltration occurs into the epidermis. The second stage is the verrucous stage, where verrucous hyperkeratotic lesions develop and disappear that leads to hyperpigmented stage, where areas of hyperpigmentations are left behind due to melanin accumulation. The fourth stage is the atrophic stage, where the areas that follow the lines of Blaschko generally disappear by the second decade, but adults may still show some areas of dermal scarring with lack of hair follicle. The symptoms of IP include developmental anomalies of eyes, teeth, and brain, skin eruption along the lines of Blaschko, and skewed inactivation of X chromosome. It's a very striking feature that is absorbed in peripheral blood cells of female patients. This skewing reflects an efficient mechanism of counter-selection against cells that express the mutated X chromosome.